Tay-Sachs disease. What is it? Tay-Sachs is a disease that is usually found in young children, although there are rare juvenile and adult cases. Symptoms appear in seemingly healthy infants at approximately 3 to 6 months of age. It is always fatal. Primary symptoms include rapidly declining development, weakening of muscles, and loss of motor skills. These are followed by increased startle reflex, blindness, deafness, inability to swallow or chew, mental retardation, paralysis, and dementia. This is followed by death at age 4 or 5. How does this happen? It starts as a defective gene on chromosome 15. It affects the body's production of hexosammonitis, which is a very important protein that breaks down a chemical in our bodies called gangliosids. The gangliosids then build up in the brain and in time cause death. The Tay-Sachs gene is recessive, so it is possible to be a carrier. For a child to be born with the disease, both parents must be carriers. If 2 carriers have a baby, there is a 50% chance of the child being a carrier, 25% chance that the child has no disease at all, and a 25% chance of the child developing the disease. Diagnosis can be made by checking parents for the Tay-Sachs gene or by taking an enzyme analysis to check the levels of hexosammonitis in the child. An eye examination can be made to reveal a cherry red spot in the macula at the back of the eye. This is a telltale for a genetic disease. There are medications available to ease the worst of the symptoms. Proper nutrition and exercise can slow the deterioration of muscles, but the prognosis will always be death by age 4 or 5. Although there is no cure for Tay-Sachs as of yet, doctors and scientists are experimenting with genetic therapy in hopes to slow the progression of genetic diseases such as Tay-Sachs.
The following images summarize gene therapy studies of direct injection of AAV vectors into the brain of GM2 gangliosidosis mice, which mimic Tay-Sachs disease in humans.
Translation. Here is a video demonstrating the effects of gene therapy in mice who have the small mammal version of Tay-Sachs disease.
These mice are severely affected and have a short lifespan of only 17 weeks. The first mouse was not treated by gene therapy, and by 16 weeks of age it has great difficulty in moving. To make this movie, the researchers used the mouse's shelter to induce the mouse to move. Difficulty in moving the hind paws becomes more apparent when the mouse is suspended by the tail. Unlike a normal mouse, the GM2 mouse is unable to bring its hind paws close to its body. The second movie is of a 33-week-old mouse treated by direct injection of AAV vector into one side of the brain only, resulting in much better function on the left side of the mouse's body than on the right side. Also, note that survival of this mouse treated by a single injection is approximately twice as long as an untreated mouse. The third movie is of a 1-year-old mouse treated by 4 injections of AAV vector into both sides of the brain at 4 weeks of age. This AAV-treated mouse is capable of moving without great difficulty and shows interest in exploring a new environment. Although movement is not normal, this mouse is alive at 1 year of age, approximately 3 times older than untreated mice. In the tail suspension test, the AAV-treated mouse retracts its hind paws, as observed in normal mice. The preceding images were provided by Drs. Begonia Cachon-Gonzalez and Timothy Cox of Cambridge University and the Tay-Sachs Gene Therapy Consortium. The goal of the consortium is to develop a gene therapy strategy suitable for human clinical trials by the year 2010.
It seems that there is a hopeful future in developing a cure for Tay-Sachs. It is important for us to be educated and to understand the complexity and importance of genetics and genetic disorders. Understanding the disorders will help raise awareness and therefore raise fundings for diseases like Tay-Sachs, which in turn will hopefully bring us closer to a cure for all genetic disorders.